Welcome to Synaptic, our podcast that investigates the people, the research, and the challenges of the neuroscience field. That's right, the show has broadened. We will still cover autism, but now our guests will include all of neuroscience. I host Synaptic, and my name is Brady Huggett. For this show, for episode 7, let's start with the Green Line in Beirut, Lebanon. After the start of the Lebanese Civil War in 1975, Beirut was divided into West and East. West Beirut was mainly Muslim and had both secular and Muslim militias. East Beirut was mainly Christian and had mostly Christian militias. These groups set up snipers and barricades along the divide that split Beirut, and in the dead zone between these factions, where no one lived and people were rarely seen, slowly and steadily a rich green foliage grew. And from that, this strip of demarcation became known as the Green Line. Now, people did cross from one side to the other through specific channels, to visit family members who lived just blocks away on the other side, or to buy bread, or to talk with friends. Militias stood at either end of these crossing points, monitoring who passed through. This, of course, made the journey dangerous. Kidnappings and executions occurred regularly over the years along the checkpoints, sometimes happening in response to each other. You killed one of ours, we'll kill one of yours, that sort of thing. But for our purposes, one night in 1984, a 17-year-old boy named Ardem Patapoutian crossed from West Beirut, where he lived, into East Beirut to spend the night with a friend. That's our guest for today, Ardem Patapoutian. Ardem and his family were Armenian, and Armenians were considered neutral in the war. But when he crossed back into West Beirut the next day, he was stopped by armed militia and held for several hours. Eventually he was let go, but when he got home and was safe again, he realized, I can't live like this. And so he began to think of ways to change his life. We talked about that on this podcast. We also talked about his journey to America and his experiences as a new immigrant in Los Angeles and the loneliness of that. And of course, we talked about his scientific journey through his discoveries working with the peripheral nervous system and proprioception, all the way to winning the Nobel Prize in 2021. All that is coming up. Now, I visited Ardem at his office in the Scripps Research Institute, about 15 miles north of San Diego. We cleared a space on his desk, and I set up the mics. And we took a long look at his office phone, right next to my recording equipment, before deciding to leave it where it was. No one calls me anyway, he said, which I found hard to believe, but actually no one did call during the interview. But there was a fan outside his window, so you can hear that a little bit in the background. I recorded him on September 22nd, 2023, an overcast but nice morning in Torrey Pines. And it was a wonderful interview, I thought. He's a gracious person and patient, or anyway, he was with me. Let's begin here, where I'm asking him how long he's been in California. This should be all you need, so here is your episode of Synaptic with Ardem Patapoutian, starting right now. You've been here in California for like 30 years plus, right, at this point?
That's right.
Yeah. And I mean, I've done some research, of course, so I know that you actually did not grow up in California.
That's right as well.
Yeah, and you were born, I think, in Beirut, yeah?
Yep. So I'm of Armenian origin, and I was born in Beirut, Lebanon, and I was there for 18 years before I came to California.
Yeah. So this was during, well, let's see. So the Lebanese Civil War started in 75.
That's exactly right. Yeah. So I was 8 years old. I was born in 67. So, I mean, Lebanon was a beautiful country,
yeah.
But most of my memories are, post-Civil War, which is kind of sad. And yeah, so I guess one message is if you can survive the war in Lebanon, you can survive science.
Yeah, exactly. So you're growing up. Did you have an interest in science when you were young?
Not too much. I mean, a little bit, but I wasn't one of those kids that, I knew I was going to be a scientist when I was young at all. My parents had quote-unquote brainwashed me that I wanted to become a medical doctor.
Well, I mean, that's science in a way, yeah?
That's science in a way. But I think mainly for selfish reasons. They wanted a family doctor.
Yeah.
And my sister and brother were both very scared of, cockroaches and stuff, and I was the tough one that would go and get rid of them. And so they're like, okay, you're the medicine guy. So that was my initial link to biomedical research.
Where do you fall with the siblings? You're the oldest?
I'm the youngest.
The youngest and the toughest then.
In some ways.
In some way. Okay, so you're growing up. When the Civil War happens, when it starts, does your family think, well, what do we do? What are we going to do? Are we going to try to live through this? I mean, I know that obviously people left the country then.
Yeah, so it's a bit complicated because of the Armenian origin. So we lived in an Armenian bubble. So my parents were born in Lebanon, but their grandparents, of course, are the product of the migration because of the Armenian genocide from modern Turkey to Lebanon.
Yep.
So although we were Lebanese, we were, in a way, both acted and treated like second-class citizens. So, I mean, we love Lebanon, love lots of things about Lebanon, but that's, homeland is Armenia still. So that complicates the equation of what you're asking is, maybe if you felt and were Arabic and you would have hung out more. But a lot of Armenians, I think, were like, okay, as you said, do we stay or do we leave? And it took us a long time to leave. So after 12 years of being in the Civil War, but we finally left. Of course, many people, Armenian and non-Armenians, left Lebanon. And yeah, it was an interesting but tough childhood.
Yeah, I mean, do you know who Nubar Afeyan is?
Of course, yeah.
Right, so I talked to him, and he was telling me, right, so your backgrounds are very similar, actually.
Very,
Armenian,
very similar.
Growing up in Beirut.
Yeah.
And he was telling me that, the buildings were coming down. There were dead people in the street, their bodies in the street, and, that was part of his childhood, too. And you're saying that sounds like that was part of yours.
Oh, absolutely. I mean, whenever there were bombs, we used to all go to the shelter downstairs, and, it's just some really scary stuff. I was even kidnapped by some militias for a few hours crossing West and East Beirut. That was actually the event that made me say, okay, I got to get out of here.
Just you were kidnapped?
Yes.
How old were you?
I was about 17.
So tell me what happened.
So I was, there's West Beirut and East Beirut.
Yeah.
West Beirut is mainly Muslim. East Beirut is mainly Christians at that time. Now that doesn't exist. And Armenians are Christian, but because we were pretty neutral in the war, we were pretty much the only Christians that could go back and forth as we wished. But I stayed the night with a friend in East Beirut, and I was walking across the border to West Beirut when I heard, gunshots, and I started running, and some militia folks who were, saw this 17-year-old running towards them, and so they stopped me, asked for my ID, and in a tragic way, in all Lebanese IDs, your religion is written on it, and so they saw Christians, so they were a bit, some problematic issues there. But I think after a few hours, they realized I was not dangerous, and they let me go. But it was a huge scare for me.
Oh, yeah. And so as you're running, you say they confronted you. I mean, guns drawn, they confronted you, stop, the whole
absolutely.
thing.
And so I think when I got home, that's when I said, okay,
I can't live like this.
I can't live like this.
Yeah.
I think the bombs are not personal, like they fall and you hide, but when it's just personally, you get held like that. It's a different effect on your psyche.
Oh, for sure. Yeah. All right. So you're growing up in this tumultuous childhood where the city is literally under siege at times, and you still think you want to be a doctor all the way through, your school.
I was actually at American University of Beirut, which is a wonderful university with an American affiliation, and I was at that time pre-med student there. I did 1 year at the American University of Beirut before
chemistry.
That's right.
Yeah. Oh, and thinking I'm going to use this to build a career as a physician.
Yep, that's right.
And then after the kidnapping, you thought, I can't stay here.
Yes.
So how did your whole family leave with you? You all emigrated at once?
So it just so happened that my mom's sister was a citizen of United States. So they had applied for us to get a green card, which got delayed and for a long time. But finally, just about that time, we received positive response for that as well. So we were able to come in as immigrants as with a green card.
Whole family? Brother, sister?
I came first. My sister never came. My brother was too old to get the green card because he was above 18 years old at the time. And after a year that I came, my parents came after that.
So you were first?
I was first with my brother, who went directly to graduate school, but he wasn't an immigrant. He was on a student visa.
All right, so then you're first, and is your plan to continue medical school? You're leading up to medical school.
Financially, we're not very well off. And so the first thing I did when I came to California is that I wanted to go to a public school, and you can't qualify for reduced tuition till you're a resident. So I had to live in California for 1 year before going to college. So the first year I just worked at a few jobs, minimum wage, and became a resident. And that was, despite all the difficulties in Lebanon, that was the toughest year, I think.
I'm sure.
So
you left your country behind, your family behind.
Yeah.
But you did
all
have
alone,
well, you had a relation in...
So my brother was in town for a few months. We didn't have a car, so he lived in... We lived 20 miles away, but we hardly saw each other. But pretty soon we got a car and started living together, and that was a huge relief.
When you were living separately, were you paying rent someplace, or what was happening?
Yeah, so I bought this tiny... I was renting in this tiny apartment and working at an Armenian newspaper doing odd jobs. And one of the things I remember actually is that I didn't have too much money to buy furniture, so I just had a makeshift bed, but I was going crazy in the silence of this apartment by myself. So I went and bought this black and white TV that I still have. I've kept it. It was $60, I remember.
I mean, with like the rabbit ears on top.
Yeah, exactly. This tiny black and white 12-inch TV. And there were color TVs at that point, but I just couldn't afford it. And I would just have it on all the time just to keep me company, because it was just a kind of a lonely time.
Anything, news, sitcoms, whatever.
Reruns of Taxi.
Oh, yeah. Man, that's... Okay, so you're living there by yourself. You're working... Were you a writer at the newspaper? You're just helping put the newspaper out?
It's so bizarre to say this. My English was terrible. I mean, I knew theoretical English, but I had a hard time understanding people because I thought they were talking so fast. But I was put in as the English section editor, which is so ridiculous. But I also helped. Every Tuesday we used to, print the labels on the thing, go to the post office, and drop the newspaper. It was a weekly. Yeah. I even got to write the horoscopes in the newspaper because the editor saw some older lady at an event, and she told him that I'm reading the same thing every few months, the same horoscopes, and how's that possible? And he came and said, "Oh, that's embarrassing. We need new material, so we should write them." So
what do you know?
So I started I knew nothing about it, and I started making this stuff up, and I was having fun with it because I would write a specific one to a new friend of mine and say, "Hey, read your horoscope," and he would come back and say, "I read it. It makes no sense. Why do you think this is relevant to me?" And then I found out that the printer just would cut and randomly put them. So I was specifically writing some advice for someone born in certain month, and she would just randomly put them everywhere.
But even what you were specifically writing for someone born in a month, you're just making that up.
Yeah.
Like this week you should look out for good luck in that. Oh my God.
Yes. So I apologize for all those people who follow this and think it's some kind of science.
So you would have to do 12 of those every issue for the months.
Yes.
Right? Oh my God. That sounds amazing.
That was the most fun, actually.
Yeah. And this paper is published for the Armenian diaspora.
Community it's all over the U.S.
Oh, okay.
Yeah, it goes actually to Boston, New York, L.A.
Wow.
Yeah.
Well, I mean, you provided. I mean, so this horoscope was going out all over the country.
Yeah, absolutely.
I mean, not that I've believed in horoscopes before, but I definitely don't now after hearing that story. All right, so that was one job that you had.
Yep.
Just to get the money for the rent while you established residency.
Yep, absolutely.
And about a year later, you could apply to schools, and you applied.
Yes.
Did you apply widely or just UCLA?
I applied to just a couple of schools in LA, UCLA and Cal State Northridge, which is another wing of public school in California system. And the idea is if I wasn't going to get into any of them, I would just go to community college for a year and then transfer. But I was very fortunate to be accepted at UCLA. Interestingly, the Cal State Northridge, that was my kind of safety school, didn't accept me. But anyways, it worked out great. I went to UCLA and
all right, so I mean, obviously there's a huge culture shock coming from Beirut to LA, right? But also, this is very difficult. You're by yourself. You're lonely. It sounds like a little bit of a language barrier, a language wall that you have to deal with as well. And then, but when you start school, you're thrown in with all these other undergrads who are, probably mostly Americans. Yeah.
That's right.
Yeah. Was it a struggle to blend in with them?
It was. I think for the first 3-4 years that I came to LA, it was a tough struggle, and that's where the Armenian community was so essential, because I was part of the Armenian Student Association at UCLA. That was my kind of social group at the time. Till I started working in a lab, that kind of a little bit replaced it. But at this time, my parents were in LA, so I actually moved in with them, and I was commuting to UCLA, 30 miles, 20 miles away from home in North Hollywood.
So they moved over, a year later. They rented a place.
Yeah.
You moved in with them.
Yes.
Did your brother? No, he was.
He was already going off to graduate school, so I think he was living on USC campus. So.
Okay. Yeah. As you're at UCLA, you're still thinking, well, I'm working towards being a physician.
That's right. That's right.
But somehow that changed.
That changed.
What happened?
So it was interesting. UCLA is a very large public school. And so my classes in biology were 100 to 400 students. And I just realized that I need great letters of recommendation to go to medical school. And I wasn't getting to know any of the professors. So my idea was to work in a lab just simply to get a letter of recommendation. And I thought I hated research because I really disliked lab courses, both in biology and chemistry. I thought they were boring, not creative, and stressful, because
what if it failed?
What if it failed and you had these expectations, it never worked out, you had to explain why. There's no joy of discovery, which later, of course, I found that's the whole point of working
Science.
in a lab.
Right.
And
but for you, as an undergraduate, it's sort of like, here's the steps that you do, and you should get this response.
Yeah.
And write it up when you're done. If you don't, you have to explain why, where your mistake. Okay.
Which I found no joy in. And so I started, there was no email. So this is, what year is this? 1988. So I started calling a bunch of professors and saying, I want to volunteer to work in your lab. And literally got 10 negative responses. And the last one was also going to be, she's like, no, this is Judy Leniel, I call it. No, I don't think we need it. And then right before hanging up, she said, what's your GPA? And it was 3.7, which is respectable, but not super amazing. And she's like, come talk to me tomorrow. And that was it. She introduced me to 2 graduate students to help them out. And literally, I fell in love with the whole culture and doing science.
Because of what they were doing? You were watching them, and they had joy for science, and you picked up on that? Or
Absolutely. So I think it was a dual thing of the actual science, which is, when they described to me that they found this new gene that controls how Drosophila goes from a single fertilized egg to this patterned embryo. And they were trying to find out how it does it. And I was sequencing the gene for the first time. So it was just super exciting. And on top of that, later on, I've always said I found my tribe in people who do biomedical research. And these were, to me, all super interesting international group of young scientists. One was from Boston, one was from Iceland, someone was from Italy, and they were all drinking fancy beers as happy hours on Friday. I'm like, what is this? This is great.
Yeah.
And then I found out that you can go to graduate school, and not only you don't have to pay tuition, you get a stipend.
Yeah.
And for me, that was it. It's like I get to do this fun thing called research, and on top of it, I get paid. This is heaven. So I kind of decided to switch from wanting to go to medical school to graduate school. So I got some pushback actually from my parents.
I was going to ask, what did they say?
They weren't very happy about this, just because they didn't know anything about research and if it was a stable job or if you would get a job, which was actually a correct concern in some ways. And my brother convinced me that I don't want to make this decision after 4 years thinking I'm going to go to medical school, that I should take the MCAT and the GRE and apply to both, and after I got in, just make a sane decision that I have an option, but I'm not doing this because I'm being lazy and I don't want to go into medical school. And I kind of did that, and it all worked out. So.
Oh, you took the MCAT, you did
Yeah.
And did you apply to schools?
I applied to schools. I did some MD PhDs thinking, this is in between the two. I didn't get into any of those. I got into UC Irvine Medical School, but I decided to go to Caltech, still in Los Angeles for PhD.
Okay. So your parents make peace with this, and you decide, well, I'm going to go into research, right? Yeah. And then, so Caltech.
Yep, not too far.
Yeah, not too far, actually, very, very close. And did you know what you wanted to study there?
I think I was very interested in developmental biology, and I did shift. I went thinking I was going to study transcriptional regulation, but then really got interested in this lab of Barbara Wald, that they were, for the first time, figuring out, kind of very similar to what I was doing in flies, but in a mouse system. The same question of how does a fertilized egg then decide to become a neuron or a muscle cell. And they had found these transcriptional regulators called the MyoD family that had the incredible ability that you put it in a undefined, undifferentiated fibroblast, and this one gene, the expression, would be sufficient to make this into a muscle cell. So this kind of became a new passion, and that's what I spent 5 years at Caltech studying. Yeah,
and then so when that finished, you went and got your postdoc. But I think also you met your wife at Caltech.
That's right.
Was that in Barbara's lab? Or?
Yeah, so she was, when I was a first-year graduate student, she was already a senior undergrad, and she did work in the same lab. And
she had the same excitement for...
Yeah, absolutely. We both were studying the same thing, and so that was fantastic. Of course, we've been together since then, and it's been a very excellent experience.
Yeah. When did you get married?
When I was a postdoc, and she just finished her PhD. So after she graduated, she moved to Berkeley, and so we did a 3-year of long-distance relationship, and then I moved to postdoc at UCSF, and then so we were back together, so and that's when we got married.
3 years of your PhD, she was in
Berkeley.
Right.
That's right.
And you kept it up, and then you went to, So you finished your PhD, and you're looking for a postdoc. And were you looking specifically for one area or one lab?
Well, I was looking at the Bay Area because that's where
Nancy
was, and to be reunited in a way. And so, and I made a decision at that time either to go into neuroscience or immunology. And the idea was that both of those fields were, I think, very exciting things were happening there. And both had, in addition to academic interest, translational promise. And I tried to read papers in both fields, and I thought that immunology was even more jargony than neuroscience, and so I decided to go into neuroscience. But it is intimidating to get into the study of the brain from when you don't have any education in it. And so my plan was to go into the peripheral nervous system, where these neurons sense, touch, pain, temperature. It's a simpler system outside the brain. And the whole idea was to ease into it, so do a postdoc there and then move into the brain.
Oh, I see.
That was my plan.
Ah, okay.
And 28 years later, I'm still in the peripheral nervous system.
Well, I get this, like the brain being this, even today, this like vast unknown world, right? We're really still trying to understand it. And you thought, well, I mean, but you couldn't have thought that was beyond your means, right?
I was a bit intimidated, for sure. I think you got to understand that it's not just the complexity of the brain. All the tools to study the brain, which is, electrophysiology, recording of neurons and stuff. So if you haven't done that, it's a completely different kind of concept, and it could be intimidating to a PhD student.
Oh, sure, yeah. So you're thinking, well, if I can study this peripheral part of the neuroscience, I can figure out the tools, and then later I can apply it to the brain if I want to.
Yeah.
Okay, good.
Yeah.
Okay, so then you studied under Louis Reichard, I think,
That's
at
right.
UCSF. Okay, so what were you looking at in your postdoc?
So Lou was involved in many different fields of neuroscience. One of them was this family of proteins called neurotrophins, and they're amazing molecules where they're kind of growth factors for neurons. And the whole idea was that you make too many neurons, and kind of survival of the fittest kind of way, only a few of them survive that get to the right target, get the right growth factors. And these were these neurotrophins, so that some peripheral neurons, for example, express the receptor and send out projections to the skin, and if they got enough neurotrophin, they would survive, the rest would die away. And so it was kind of very Darwinian idea at the cellular level. And so how these neurotrophins were regulated and what was the consequence of not having them in vivo was kind of part of my postdoctoral work.
So that takes you to almost 2000,
That's
I think, right? Yeah. So you finished your postdoc, and now, well, I mean, you have to decide what's next for your career. You're married at this point, right?
That's right.
Yeah. So how did you and Nancy decide what to do next?
So, at first we were thinking that I should wait, because I had kind of done enough postdoc work to be able to get a job, but we thought we would wait and look for jobs together. But people strongly suggested that if you have a good paper published as a postdoc, you don't want to wait 2-3 more years, because if you're not productive after that, people will question it. And Nancy very generously encouraged me to look for a position right then and there, and said, we'll figure it out. And so I mainly applied to academic places, but it was the middle of the year, so I did a small search. People in my lab now apply to 30-40 places. I only applied to 3 places. And one of them was this very unique position here in San Diego, where Pete Schultz, who has become my mentor, has started a new institute called the Genomics Institute for Novartis Research Foundation, short for GNF. And this is an institute that the Swiss pharmaceutical company has funded, but it was not going to take active role in managing it. And we were just going to explore the new genomic data, and they might get some interesting targets for patenting and working on it. So it was a fantastic environment in a way to do research in a slightly different way.
Right, because you're, number one, pharma usually, and Novartis does have some money. So you should have the money that you need to do the work that you want. And whatever you want to look at, they're okay with, as long as they get to sort of maybe pick and choose if anything does sort of strike their fancy.
That's right. At the same time, I had interviewed and had an offer from Columbia University in New York, and so I kind of told these guys in San Diego that this is great, but the idea was like a Bell Labs. You only have 3 people in your lab, but you do whatever you want. It's all paid for. And so they managed to say, okay, we'll give you a joint offer with Scripps, which Pete Schultz was involved. And so I got this wonderful dual affiliation. I was 50% academic Scripps lab, and 50% my was at GNF. And so started 2 physically labs next to each other.
I didn't know that. And the GNF one is very small.
Yeah. But they had lots of core facilities where you could collaborate and get stuff done. So it was very, very incredible.
When you were saying, well, I also have this offer at Columbia, what you're saying was, I want a bigger group to collaborate with?
Yes, I think in biology, having a good sized lab is important. And I always thought that, 8 to 16 is a good number. Because with only 3 people, it's just very tough to be very productive. Some people do it, but it's great to have more. Also, as I've learned in my career, is that if you only have 3 people and 2 of them leave at the same time, you lose all the connection with the ongoing work, and so the new people have no one to learn from. So I think having a good sized lab is important for that reason as well.
Almost
where
like
the
protective.
turnover is not as painful.
You said earlier that if you're coming out of your postdoc and you're looking for work and you've had a good paper, you should kind of strike while the iron's hot. What paper had you published that?
We had published a paper in Science that showed that this other pathway, the Wnt pathway, controlled one of the neurotrophin, called neurotrophin 3, which was, again, very much required for survival of a special population of neurons, including proprioceptive neurons, in the peripheral sensory neurons.
Okay.
So it was just connecting two different pathways together, because I told you neurotrophins were very important in regulating how many numbers of neurons survive, but almost nothing was known about how neurotrophins themselves are regulated. And this was kind of one of the first clues of how that gets regulated.
Okay. So you're setting up these dual labs. Are you still affiliated with the Novartis lab? You still are?
No, I'm not, actually.
So what happened? How did you become fully with Scripps?
So after Howard Hughes Medical Institute investigator, I became an investigator. That affiliation became a little bit more complicated. And so as the director change of Novartis, Martin Seidel and Pete Schultz were very excited to keep this collaboration that I think has served both institutes very well. The new institute had thought that was more problematic in keeping that affiliation, and so it kind of came to an end.
Because these ties to industry, right?
Yeah. Of course, after the Nobel, the head of Novartis Research told me that he's so upset that that relationship didn't continue till the end, because they could have claimed the Novartis researcher becoming
the Nobel. Yeah, my God, yeah. Okay, so you're building your lab at Scripps then. I'm solely doing that, and eventually you just mentioned the Nobel. You do win a Nobel. So what work were you doing that led you up to where we are now? Like you won the Nobel in 2021.
That's right. So one of the things I think realized when I started my own position is that I was very interested in this developmental questions of how these sensory neurons become different. How does one neuron decide to sense cold while another senses pain, while another senses light touch? Very interesting question. But it was just this idea that, what's even more interesting is how these neurons actually sense warm, cold, touch, and pain, because this is something that all neurons have to develop and become different. But these...
And they're specific for each?
Yes.
Oh, okay.
Yes. But how do these neurons actually sense physical stimuli? Pressure is energy, temperature is energy. How do you translate these physical stimuli into an electrical signal that neurons understand?
Yeah.
Right? There's nothing known about that. When I actually started my lab in 2000, just a couple of years before, David Julius's lab had identified the first heat and capsaicin activated ion channel, which turned out to be the channel that you use to sense noxious heat. And so we took a genomics approach to find more of these. And the first 10 years of my independent work, I made this sudden change, which almost no one does, because everybody assumes that what you did as a postdoc, you have to take that with you and expand on that.
Yep.
I mean, it was kind of related. I was working on the same neurons, but the question became very different. And so my lab identified the first cold activated ion channel, TRPM8, which also senses menthol, which has this cooling effect. TRPA1, which senses many pungent compounds like wasabi and garlic, as well as other ion channels. And this was, work that was also done in David Julius's lab, my lab, and many others. But it was a very exciting time where we were finding the same family of ion channels, these transient receptor potential ion channels, each tuned to very different modalities that gives us the sense of both chemical and thermal responses to these stimuli.
But the idea that there's something for sensing wasabi, right, is so specific. It isn't specific to wasabi. I understand that. There's other things that it can sense. But how have neurons evolved to be that specific?
Yeah, it's one of the most fascinating stories my lab has been involved in. So it actually, most receptors, they sense ligands by this old-fashioned lock and key mechanism. So there's a pocket on the receptor, the ligand fits, conformational change activates downstream pathways. So TRPA1 is evolved to do something completely different, and that is it senses reactive chemicals. So actually wasabi and the active ingredient in garlic don't have any similarity in their shapes. All they do is react with cysteines, so they are reactive chemicals. So TRPA1 has evolved to sense reactive chemicals that it senses through its cysteines and sends the message. And the idea is it's evolved to recognize damaging stimuli. So wasabi might not be that damaging, but it kind of fits into that little niche of molecular biology, and that's why it feels like burning. Most animals would think of it as pain. We humans, being unusual, have grown to love it.
So it's the cysteine that is the commonality there.
That's right.
That is so fascinating.
That's why all these ligands that have no shape similarity can all activate TRPA1. And when we did screens to find agonists of TRPA1, usually you would, at Novartis, we would screen 2 million compound libraries and find a handful, maybe 100 activators. For TRPA1, 10% of the compounds would activate it almost, because there's lots of reactive chemicals out there.
So I'm also, when you were thinking about your research, right? Did you just wonder one day, you're like, how is it that, because it's such a simple question, and it's never occurred to me until I started looking at your research. It's like, how is it that when I put my hand against the wall, this outward pressure creates a current that then tells my body what's happening? Did you just wonder one day how this works?
So I think the conscious decision in 2000 when I started my lab to focus on these receptors had that in mind already. And so there was temperature, there's chemical, and there's pressure sensing. Pressure sensing was completely unknown. There was nothing known at the time. And as I said, the first 10 years we kind of worked on these temperature sensors. There was always in the back of our mind of the elephant in the room is this mechanosensors. Not only they weren't known in touch and pain sensation, we didn't know how muscles or blood pressure, all having to do with pressure, is regulated. So that was something we were very interested in, but it took us about 8-9 years to start focusing on it because we were so busy working on the temperature sensors, which I think are also very interesting, of course. But it's just in science, sometimes you have to look at a field and say, okay, I think the main big questions of temperature sensing are now maybe solved, and the field is getting into more details. And I should say this is a very subjective analysis. There are still people doing excellent work on those sensors. But for me, what I was interested in them was kind of answered already, and so then we made a conscious effort to say, okay, let's not focus on pressure sensing. Right,
you're like, here's a new challenge. Let's figure this out. Okay, and this, I think, if I'm not mistaken, this is piezo 1 and piezo 2,
That's right.
Right, so tell me about working with that.
Yeah. So Bertrand Coste, this postdoctoral fellow, came to the lab who had experience recording from these channels, and so it's kind of a very simple assay where it's whole cell electrophysiology. You're recording currents in the cells while you actually, with a glass probe, poking the cell. And if the cell expresses a mechanically activated channel, then just simply gentle poking of the cell will elicit a current.
Wall. Okay.
The plasma membrane of a hex cell, let's say. And so initially, we were trying to find this from this DRG, peripheral nervous system neurons that we've been working on for a long time. His PhD actually had recorded from these neurons and found these beautiful mechanically activated currents. But very soon we realized that these are very heterogeneous cells. They're difficult to work with. They don't divide. You have to culture them. And trying to find it from them was close to impossible with the current techniques. So there we made a decision that made all the difference. And in science, some people say very correctly, and I agree, you should pick the most reductionist approach to a question that's still meaningful. And
as your first question.
As your first question. Yeah. And so if our interest was to find a mechanically activated channel, why not find it from easiest system, which in this case would be a cell line that's homogeneous. You can divide and grow it in a petri dish as much as you want, find it from there, and then you go back and say, where's this relevant in vivo? Is it in the RG neurons? Is it in heart muscle? Is it in other pressure sensing system? Maybe hearing. And this was the key. So because then Bertrand just screened cell lines, easy to work with cell lines, and found that this one of them that's heavily used in neuroscience research called Neuro2A cells actually had mechanically activated currents very similar to what was found in the RGs. And once he picked that, he used at that time new technique called RNAi screens, which you can knock down one RNA at a time. And so he made a list of genes that are expressed in Neuro2A and not in other cells, but could be ion channels, which mainly means that it has to have multiple transmembrane domains. Somehow made them into a list, and one by one started knocking them out to see if you knock down gene X, would this mechanically activated current go away. It was still not easy because it was such a low throughput screen. So to test each candidate, it took him about 2 to 3 days to knock it down. Just think about it, that his list was 300 genes long. And so we were ordering primers to knock down 30 at a time. So the first 30 all negative, the second 30 all negative. We were starting to get very nervous for his career, because as a postdoc you can't just have negative data.
But so if you screen 30 and you get a positive, then you go one by one, is that right?
Well, if you get a positive, you stop, because it means you found it.
But in that 30, I see. Oh, okay, I'm sorry.
So what he's doing is knocking genes one at a time and seeing that the mechanically activated currents are completely normal. So it's the wrong candidate. And so 30, 60, and I think we ordered the next 30 saying, after this we probably should find you another project, because he's been there 2 years already and with no data. But the 72nd one was it. So when he knocked down this gene called FAM38A, which is unknown family 38 member A, he saw that this mechanically activated current almost disappeared. So that meant that this gene is required for this current. That was not the end of the story, but it was a huge eureka moment in a way for him and me and the whole lab. So later on, of course, when the next experiment you do is you get the full-length cDNA of this gene and then transfect it in a completely naive cell, and he saw massive mechanically activated currents. So that meant that this gene is both necessary and sufficient to induce mechanically activated currents, which in genetics, that's what you want to believe that this is it. Of course, it took many more experiments to completely convince us. But that was the beginning of the big discovery.
And so and you're publishing on this all along the way, I assume?
It took a little bit more experiments than I was just suggested to even publish the first one.
Okay. But eventually you begin to carve out space in this field, and people begin to take notice. And you're working with David Julius at this time?
No, we were, so honestly, we were competitors when we were working on the TRP channel. So this reactive chemical of TRPA1 that I mentioned, I thought it was such a bizarre and unique discovery. I couldn't believe it that we were finding out. And yet David had found the same thing, and we kept publishing a few weeks apart in competing journals. So it was But
were you communicating with each other, or you would just see the paper and you go, well, he's done it too? At what point did you just go, well, we should stop, maybe we should just work together?
That's complicated if you have exactly the same interests. And so we never got to work together. But I think my big advice to people coming up in the field is, I'm being completely honest here, is that it was a competitive environment between David's lab and my lab. And to me, that was a negative feeling that we were competing. But looking back now, I feel like we really, at least from my perspective, we enabled each other. So I think there's this negative thinking of competition, but it's actually such a positive thing. The way to think about it is that we're finding out these novel genes and novel mechanisms, and how wonderful it is that within a few months you find that someone else has done exactly the same thing and found the same results. And sometimes when you don't find the same results, you keep looking at it and find out what's really happening. So this idea of competition as a bad thing is something I experienced during the process, but now look at it as a wonderful thing.
Meaning that it spurred you on?
That's right.
Yeah, it kept you on your toes.
Kept you on your toes, but at the same time, as I said, science is not real till it's replicated by others. And we were getting instant gratification in a way.
That you're right.
That you're right. When you publish a few weeks apart in completely independently, that's instant verification, and it's a good thing for science.
Yeah, we're on the right path is what that means. Yeah.
Absolutely.
Okay. So I'm going to jump ahead. So
yes.
The Nobel Prize happens in 2021.
Mm-hmm.
And some of this is out there, so we don't need to get into all of it. But I mean, this was a pandemic year, right?
Mm-hmm.
And I know that, just briefly tell me, so the way that it works, of course, is the Nobel committee decides, and then they call the people. And if you're in the United States, for instance, it's usually very early in the morning because of the time difference. And they were trying to do that with you, but couldn't get through. Just tell me that story.
That's right. So, I mean, like most normal people, I have do not disturb on my iPhone,
Mm-hmm.
which means you can't get through when I'm sleeping.
Yeah.
And so they were calling at 2 a.m., couldn't reach me, and they have someone there, I found out later, who specializes in this emergency situations to find someone close to you. And so they were Googling Patapoutian, California, and they found this other number that they called. And it was my father, who's 94 years old. And his landline rang, and he answered, and he yelled at them first of why they're calling so late at night. But
Who is this?
Who is this?
Yeah.
Why are you bothering me? What are you selling? But I think he quickly realized what was going on, and then he was able to contact me.
Because he's on your
Because
safe
he's
list
on my
or whatever.
safe list.
Yeah.
And when I looked at my voicemails, there's 3 missed calls from Stockholm, so I kind of figured out what's going on, and so I was able to connect with the head, Thomas Perlman, the head of the committee, just before he announced it publicly, and so I was able to watch it on my computer, the announcement.
So 2 things. One is, do you know when you're in the running? Do you know? I've always wondered this. Does the Nobel say? I don't think they do, right? They don't say there's like 6 researchers we're considering this year.
No, they don't.
So you had no idea?
No, I did have an idea, but it's a complicated answer, so bear with me.
Mm-hmm.
People tell you, people tell you either that, I think this is
your year.
Not this your year. You don't know that. They say you're in the consideration. Other people, maybe they should or shouldn't say, but they say, I've nominated you.
Okay, all right.
So you kind of know that it's in the works. You also, David and I have won a couple of prizes before that.
You won a Kavli Prize.
The Kavli. And so you kind of, it would be very naive to say, I had absolutely no idea. But you don't know the year, and also philosophically, although I knew, I've kind of put it aside. I didn't want to become one of these people who either lobbies for it
fixated
on it or consider that a goal. Didn't want any of that to happen to me. So that's why I wasn't going to undo my do not disturb that night. And completely honestly, because of the COVID that you mentioned, like many others, I thought it was going to go to something related to COVID research, either the vaccines or identifying the. And so that year to me sounded like that's obvious. Now that I know the process more, I'm not surprised that they haven't given one to that also, because the amount of research they do is mind-boggling. They take this so seriously.
The Nobel Committee.
The Nobel Committee. So afterwards they told me that they had a file on me for 15 years.
Oh, wow.
And that for COVID, I'm sure they're doing...
It takes a while,
It takes a while to make sure that they're giving it to the right discovery and to the right people. And so people have to realize it takes time.
So because of it being COVID, normally what happens is, they announce who the winners are, and then there's a press sort of... the press descend on the house, and someone's there with a camera and all that. And that wasn't going to happen because of COVID precautions. But I think they reached out to you and said, look, we're going to announce this online, and is there a way that we can get some sort of photo of you finding out or seeing this online? So what happens is you're in bed, I think, because it's still early.
2:30 a.m. Yeah.
You're in bed with your laptop watching, and your son is in bed with you, and your wife snaps this photo. And it's a great photo. It's like you have just looked up at the camera, and your face is kind of, it's not like, what do I want to say? You're not shocked because you already gotten the call,
Yeah.
but it's like a bemused tranquility or something, right? You're like, well, there it is. But your son, he's got his hand on your shoulder, and he's looking at the screen. He's like, that's my dad, right? I thought that was a very touching photo, and it got quite a bit of stir, I think, online.
Yeah. It is my favorite photo for many reasons. One of them is the look being next to Luca, my son.
Yeah.
And I always joke around that he doesn't affectionately touch me like that all the time.
You have to win a Nobel.
And I study touch sensation, so it all comes together. And, whatever you say about Twitter and social media, in the absence of direct human contact, that's how I connected to the scientific community throughout the world with that photo.
Yeah.
That was the introduction of it, as well as, the Lebanese and Armenian community who've been so proud of this. All happened through this social media and that photo. So it does have a very special place in my heart.
Yeah, I would think so. I mean, and I don't know if your wife spends a lot of time taking photos, but it was almost perfectly placed in the frame. It was really, nice.
She is very good at it.
Yeah. All right. There's a few things I want to ask
Sure.
you. And so the Nobel happens, right? And this has put an amount of notoriety on you, right? I mean, the world is sort of looking at you now, and they sort of know your name, and if they didn't before, they certainly do now. And you're etched in history, right? And it seems to me, just tell me if I'm wrong, since then you have sort of, I don't know, it's like it's almost freed you up a little bit. There are things you've said in the past that when you first came to the U.S., you're like, I kind of have this long name, Patapoutian, and maybe I should shorten it, or that you didn't want people to know that you'd gone to the American University of Beirut for some reason. And then you're like, I'm not going to do that anymore.
That's right. I think the liberating term that you used is just so apt. I think it's this. I also say things like, when I used to go to scientific conferences, I used to have a little bit of a chip on my shoulder, like an agenda, in the sense that, I should meet this person who's famous. I should get to know this person who might be useful for us to collaborate with, or something like this. And Nobel being the ultimate in external validation, I feel like I don't need that anymore. And so both whether it's conferences or in the lab, it's gone back to the original reason why I got into science, which is the joy of discovery. And it's just this wonderful feeling that I don't have to prove myself anymore, and I can just enjoy discovery, which is what we all get into science to do. But what I would love to do is to give this advice to younger folks doing in science. And of course, it's easy for me to say this after the recognition.
Right.
But I truly believe that without receiving that validation, you can have this mentality to do what I'm doing now to a certain extent. You can tell yourself that I'm not going to stress out if I don't get every grant, or if I, don't get respect from this one scientist, or if I don't get this paper accepted. Just focus on what you love about science. Again, I say this with lots of hesitation, because someone will listen to this and say, it's easy for you to say, if I don't get my next grant, I can't do science.
Right.
And I understand that. I'm not saying completely forget about practical matters. But I think even if you move the needle a little bit towards the joy of doing science, then you'll be much better scientist and happier doing it.
Yeah. All right, I want to ask this because you mentioned earlier in Beirut, the Armenians are sort of, you said, second-class citizens, as you said. And then you come to the U.S. Immigrants feel like an outsider, have to learn the language, fit in. You're again sort of like outside. Is that part of the chip that you said you had on your shoulder?
Absolutely. And the example that you gave about, which I, admitted to, is that when I first came, because of my name, and Beirut, Lebanon are just jokes about terrorism, and so I did not mention that I was at American University of Beirut for 1 year, just put UCLA graduation kind of, and but again, what I've noticed is after the Nobel, I've gotten so many comments about other immigrants who look at me and say, oh my God, there's hope for us, because I feel like I don't belong I have imposter syndrome. And then this guy that wasn't a genius biology with a pedigree of Harvard and MIT came here poor and made it, so I can do it too. I never thought of myself as being a role model in that sense, but almost got, I don't want to say forced into, because I do it gladly, but seeing that response gives me so much energy. And my whole Twitter account, I refuse to call it X, is one of the goals is to do that, is just show that I wasn't very special, and I've made it, and you can too.
Yeah.
And that seems to really resonate with people, and I love that, actually.
Yeah, I had this thought too, that like as you get older, I don't know, when you're young, maybe you're embarrassed about parts of your life. I don't have this degree, or my parents were this, or my parent was in jail, or who knows what it is. But when you get older, you realize that everybody's life is kind of got things that maybe they're not proud of, and it's fine. And
That's
you've
right.
got that reached that point where like it doesn't matter.
Yeah. And also just sharing failures or sharing all this stuff, you think sometimes is a bad thing, but people love that, and I think you'll be more appreciated and understood better if you talk about those things.
Yeah. I mean, I guess if you had, as a scientist or whatever in life, if you had just failed and never won the Nobel, it's a lot harder to put that forth. But when you've actually got the validation, as you said, look, I was, you might see me.
That's exactly right.
Yeah, right.
That's why I'm very careful in saying I don't want someone to think that, you've got the Nobel, now you're telling me I should do this and that. But I don't want to come off at that. But as I said, it's just a matter of where the needle is. It's just understanding this, dealing with it, and keeping the joy of discovery is very important for scientists, I think.
All right, two things left.
Yep.
The U.S. has had this long issue with immigration. Administration after administration has just kicked it down the road. They haven't really tackled it. And I live in New York, and our mayor has said we're having an immigration crisis. There's just, the shelters are full, right? And I'm wondering, when you talk about immigration, what your feelings are about it.
I mean, all I can say is that I've experienced it. It's very tough, but part of that, going through that difficulty, really prepares you to take on other difficulties in life. And it's not there was actually this one article, I don't remember the exact percentages, a huge percentage of people who've won the Nobel, not that that's just a very important criteria, but it's a fact, that are immigrants. And so I think there is something about it. I think
Drive.
It's something about the drive, something
Yeah.
about experiencing something and then experiencing something else, and the knowledge you get from that. I feel like even in science, forget about immigration from country, people who change fields seem to do better because they look at a new field with a new perspective, with a new frame of mind. So I think this movement, this change, whether within scientific communities or within countries, is such a great driving force for discovery, and it should be encouraged.
Final question. I also saw that you said, I think this was maybe in your comments to the Nobel Committee, but there is nothing more exciting. There's no more exciting sentence in the world than somebody standing by a microscope and saying, "You got to come look at this." You still feel that?
Absolutely. I think this comes full circle to what I was saying of now doing research for the love of discovery, and I had to sit down and think, what is it that I like the most? And I often say no to administrative jobs because I love that being in the lab. I used to travel to 12 trips a year, and I have kept that. I don't do more than that, and part of it is to have these discussions with students and postdocs, and once in a while asked to go and look at a microscope with an incredibly unexpected result. That's the biggest science joy that I still experience.
Perfect. Thank you very much.
Thank you. This was fun.
Really enjoyed that interview. I learned a lot, both about science and how to better consider my place in the world, I think. Maybe you did too. I don't know. Thank you, Ardem, for making time and allowing me to set up in your office. This podcast will be archived on spectrumnews.org and is available wherever you find podcasts: Spotify, Google, Apple. Find it and subscribe, and you'll get each episode. You can also rate and review Synaptic, which does help other people find the show. Some of the information on Beirut, the Lebanese Civil War, and the Green Line was taken from a short video produced by Timeline, which examined the work of Lebanese photographer Patrick Baz. Our theme song was written and performed by Chris Collinwood. I will talk to you on episode 8, which I've already recorded. And yeah, here comes the music to take us out. Okay, I think we're ready.
What's your background?
So, you mean like professionally?